Hello everyone, welcome to my Biology 110 presentation. Today I'm going to talk about cell transformation and synthetic water pollution detected cells made by my team. Firstly, I'm going to talk about the principle of cell transformation, and then I'm going to talk about my real-world connection, synthetic water pollution detected cells. So cell transformation is the process of transferring foreign DNA into competent cells. The competent cells are the cells that have the ability to pick up foreign DNA from the surrounding environment. The foreign DNA usually refers to the plasmid. We need to activate the normal cells into competent cells through some chemical or physical process, like putting the cells into salt solutions or deploying electrical pulses on the cells. The plasmid is usually a small circular double-stranded DNA found in the bacteria, as we can see here. They can replicate and transcript independently without the control of the cells. So in the plasmid, it usually has the origin of replication, antibiotic-resistant gene, selectable marker, and our inserted gene, which is the gene that we want to be expressed in the transformed cells. Here is the overview of the cell transformation. Basically, it is inserting the plasmid into the cells to get the transformed cells. Okay, what we did in the lab 6 is that we transform the plasmid containing ampicillin-resistant gene, the LAC-Z gene, into competent DH5 alpha cells to get the transformed cells. And the ampicillin-resistant gene and LAC-Z gene is used as selectable markers to select the already transformed cells. The importance of the cell transformation is that it is used as a tool to transform genes to cells, and the genes will be expressed because the plasmid can replicate and transcript independently without control. So we can change the characteristics of a cell, let it produce the protein that people want it to produce, or we can do some gene testing, which I will mention later. The plasmid is powerful because in this plasmid, it has 2 restriction sites that can be cut by restriction enzymes. Therefore, we can insert whatever genes we want into the plasmid to be expressed in the transformed cells. Here's the graph showing the process. The original gene will be cut by the restriction enzyme, and the plasmid will combine with our inserted genes to produce a recombined plasmid waiting to be transformed into the cells. The second part I will talk about are my real-world connection, synthetic water pollution detected cells. This relates to our IGEM competition project. So what my team did is that we synthesized cells that can produce fluoresces when interacting with copper. Therefore, we can use the fluorescence level to indicate the level of copper. We can partly show the water pollution because copper is one pollutant in water. The principle is like here. So we already have the transformed E. coli cell that has the transformed plasmid containing the operator. The operator is synthesized using synthetic biology. When the copper goes into the cell, they will deactivate the repressor, combining to the operator. Therefore, the reporter, which is the fluorescence shown gene, will be expressed normally, showing the fluorescence. So we can use the level of fluorescence to detect the level of copper. However, we still need to increase the sensitivity of the cells by keep synthesizing the operator gene and doing the experiments. We have many mutants, and we plug in the genes into the plasmid, transform it into the E. coli cell, and test its sensitivity to copper. This is only a small part of our mutants. They are muted, and we test their sensitivity according to the copper concentration. Finally, we have our best mutant that is mostly sensitive to the copper. That's all. Thank you.